next speech, that is Dr. Pînzaru from Romania. Okay. And I think the title is too long and the time is too short, so we should just start, please.
Thank you very much. It's about a new Betulin-based nanoemulsion and its efficiency in skin cancer. Thank you. Here I will resume some of our results obtained in cooperation with Medical University from Timișoara. I'm coming from Cluj. Here is Cluj, Romania. And my university implemented for the first time a trans and multidisciplinary approach to such an enterprise to combine the physical techniques of high sensitivity to try to solve something in medical field. Here I will resume only some of the results, not all because of the time limit, and I will summarize with a little brief description of Raman spectroscopy, which I know is caring for medical doctors and for the clinicians, but I'm confident that it's a very, very useful tool which, that's for sure, will reach the laboratory of the hospitals and the diagnostic laboratories in very, very short time. I will summarize some of the facts about why is important Betulin and its formulation in various skin cancers. Then I will describe a little bit about the physical, chemical properties and their application in mice models in order to see which is the effect. And finally, I will draw some conclusions regarding the importance of such a new formulation for skin cancer. In brief, about Raman spectroscopy, it's, without entering into the quantum mechanical aspects, it's just inelastic light scattering. And from this scattering, you get information on solids, liquids, gases, cells, or tissue. So it doesn't matter what kind of sample do you have. You can get extremely important information concerning structure, dynamics, in vivo, without touching the body, without touching, without cutting, without preparing, without adding chemicals, and so on. Therefore, I'm confident that Raman spectroscopy techniques deserve better attention from the medical field, and that's why I propose to highlight some of these aspects. And here in the right side, I should present in brief our expertise in these techniques since the very first equipment acquired in 1933 in our university in Bolyai to present. I would highlight that Raman spectroscopy exhibited an exponential growth concerning the application in biomedical field. Here is a time span of 20 years of the number of scientific publications in the field regarding Raman spectroscopy for the 20 years. There are basically two kinds of equipments which use the light coming from the sample to analyze them in a dispersive mode or using a microscope in the beam to analyze the samples at microscopic level. What you obtain is a spectrum which any spectroscopist can interpret and can provide valuable information about your sample. Here are some images from the laboratory of Bolyai universities. We use different kind of lasers. We can choose any wavelength we want. And the equipment is rather complicated. They are coupled. This is the main system coupled with the microscope. You can put your sample here, even living organisms. And the system is coupled with an atomic force microscope. So you can do nano description of your sample in the case you use various nanosystems which you want to integrate into a cell or tissue. And here in brief, when the laser comes to any kind of cell or tissue or whatever you have, the focusing objective is collecting back the information, and all the information you extract further is the scattered light, which is very specific for each molecular species. Therefore, the know-how in Raman spectroscopy helps a lot in understanding phenomena in vivo without interacting or modifying their natural course, without introducing new chemicals. You can just see what's going on in vivo. Of course, the weakness of this part is the lack of reproducibility when you analyze, for instance, two cells from the same culture, treating in the same way or incubating with the same nanoparticles, and you can find out that the cell from the left side is different from the cell to the right. Okay, this aspect was also covered by the prestigious Science journal, even this year. They highlighted that each cell in living condition exhibits in a very, very particular way. So it's an individual, so you should not be blamed if the cell number 1 is different from the cell number 2 in the same culture or incubated with the same nanostructures. In brief, our research was developed following a similar principle, learning by doing. We needed some expensive molecular compounds to probe on skin cancer models, because several nanograms of Betulin or its derivatives cost almost several €100. We identified natural sources. It is not our discovery, it is from ancient times recognized that betulin coming from the outer bark of the birch tree contain large amount of pentacyclic triterpenes, which act as anti-inflammatory, cancer-effective compounds, anti-aging in skin problems, and many, many other important medical properties. We discovered that their solubility is absolutely lousy, therefore we needed methods to improve the solubility, including them. We use various cyclodextrins to produce an improvement in their solubility for formulation. So we conducted theoretical vibrational spectroscopy in conjunction with theoretical methods to describe such compounds, of internalized the species into cyclodextrins, but we discovered then that they are hardly released from these systems. Therefore, we passed for other methods like formulation in nano-emulsion. And then finally to evaluate their effects on mice models. This beautiful picture comes from a Romanian forest of birch. The bark was collected from 2 different areas, and the extraction procedures were optimized. Then finally we reached 92% final compound, which showed an effective activity, and because of the other compounds up to 100% remaining, it seems that the synergic activity of the other minor compounds could help in their effectiveness. We also built up a database of various species, and we explored their content in such betulin derivatives by building a database like this, in order to provide that know-how where to get the best compound, in which time of the year, which is the best species to extract the higher amount. And looking just before this conference to see what's new in the field, we just found out after a normal Google search that someone quoted as a team of researchers from Romania having studied these anti-cancer properties. So we were very happy to find out this. The unhappy part is that no one else took the results to probe in some other cancer models. Facts about Betulin and birch bark. We use vibrational techniques to characterize these compounds in various formulations. So we formulate these compounds in different ways to see which is the best and the proper version for applying in skin cancer. This know-how about fast identifying the richest content, the best species, and the harvesting time using portable Raman techniques was also possible. So here we are with the formulations, and the latest one is a topical formulation of a nano-emulsion. An ointment was less effective, as I mentioned before, complexation in cyclodextrins, even pure or derivative of cyclodextrins, of various ramified cyclodextrins, were less successful. Here is the new prepared nano-emulsion, which was also characterized using Raman spectroscopy techniques. So here is the blank one, and here is 1 millimolar compound introduced into the emulsion. As you can see, at this concentration, there is not big deal in the changing of the spectral response. And here is the pure Betulin compound. So we can easily recognize the Betulin compound when it's in high concentration, but in this concentration, we definitely need different specific techniques for limit of detection. The small changes appeared in the region which is highlighted here. And here we characterized by transmission electron microscopy the particles, the dispersion, the dimension, and we built up a distribution of their diameter. And we observed that the nano-emulsion exhibits particles which describe better a Lorentzian fit rather than the blank one which describes a Gaussian fit, which indicate that the compound interact with the lipid bodies to provide such a better approximation of their distribution. Except these aspects, we determine the zeta potential, the hydrodynamic diameter, and we also conducted other experiments to characterize their efficiency in absorption, in transmission through membranes, but I will not talk about this. Yes. We use four groups of bulb C mice, one with early melanoma, the other group typically treated with this nano-emulsion after inducing melanoma. The third group was only solvent treated, and the fourth one was healthy. And for the first time, we applied for such a large group Raman spectroscopy techniques to see in vivo if we can make the differences and record the molecular changes associated with the effect of the treatment. The molecular changes were recorded as spectra and were integrated, were analyzed. I will not get into the details of spectral characterization. And of course, the results were compared with the standards of immunohistochemistry. So here are the three cases. We omitted the healthy one because it's similar with the solvent-treated group. And here is the Betulin-treated, and here is the cancer group. What we see from integrating the band characteristic for the changes. Yes, yes, I'm ready. This is the last one. What we see here, the treatment makes the difference between the cancer case and the health case. This is what I wanted to highlight. And here is the area of interest. It's a matter of in vivo evidence, and except in vivo evidence, we also can image the area to highlight which is the main compound responsible for this signal. Without assigning the spectral, which is not the case here for the time limit, I will just say that in vivo is possible because between two breathing under anesthesia, there is enough time to read the molecular structure. So this is, I think, the unique technique which allowed you to see in vivo without damaging the animal what's going on at the molecular level. I will get to one
thank you very
conclusion.
much. We have to stop here because we have just 10 minutes left for the two last presentations, so we cannot allow any question for this presentation. Excuse me, we have to postpone it for the coffee break in the bilateral way. Thank you very much. So we have a next speaker from